Welcome back, everyone, for another deep dive. This time we're focusing on Coenurus cerebralis, a parasitic disease that,
Yeah,
affects sheep and goats primarily.
right.
And you shared a recent study with us, and it seems like you're really interested in the genetic diversity
Yeah,
of this particular parasite.
yeah.
So this study focused on a pretty unique case.
Yeah, it
Right.
investigated the genetic makeup of multiple Coenurus cerebralis cysts that were found in a single sheep brain.
Okay, so right off the bat, they found 7 distinct cysts
Oh, yeah.
in one sheep's brain.
Uh-huh.
What is the typical number that you would expect to find?
It's an
Animal.
Finding 7 cysts in a single sheep brain is very unusual. Typically, infected sheep will present with far fewer cysts,
Okay.
often just 1 or 2.
Gotcha.
So that really highlights why this particular case is so intriguing and, worthy of a deep dive.
Right. Yeah, absolutely.
Yeah.
So how did this case even come to the researchers' attention? Like, what were the symptoms that this sheep was exhibiting?
So the sheep was showing a range of neurological symptoms,
Okay.
things like listlessness, loss of appetite, difficulty walking.
Oh, wow.
And unfortunately, these symptoms were severe enough to warrant euthanasia.
Right.
But that difficult decision did provide researchers with a valuable opportunity to examine the sheep's brain, and that's when they discovered these multiple cysts.
Wow, that's wild. It's so fascinating how one case can open up
Yeah.
so many different avenues of research. I am curious about the genetic
Yeah.
aspect of this study.
Right.
What were the researchers looking at specifically, and what makes that information so valuable?
Yeah, so the researchers focused on analyzing the MTCO1 gene in the parasite.
Okay.
This gene is located within the mitochondria of the parasite.
Mitochondria, powerhouse of the cell.
Exactly, which are responsible for energy production. And mitochondrial DNA is particularly useful for studying genetic relationships because it evolves at a faster rate
Okay.
compared to nuclear DNA and is inherited solely from the mother.
Okay, so when we're talking about these genetic relationships, are we talking about the relationship between this parasite and other parasites, or are we talking about
Yeah, so
evolutionary history?
we're looking at the relationships between different Coenurus cerebralis parasites,
Okay.
so different strains or isolates of that parasite.
Gotcha. Okay.
And by looking at the mitochondrial DNA, we can get a sense of how closely related they are
Okay.
and how they might have evolved over time.
Cool.
So it's kind of like building a family tree for the parasites.
That's really cool. The study mentioned something called haplotypes, these distinct genetic variations.
Yeah,
And it seems like they found quite a few, right?
that's right. By comparing the NTCO1 gene sequences from the cysts in the sheep
Okay.
with sequences from other studies, they identified a total of 21 different haplotypes of Coenurus cerebralis.
Wow.
But here's where things get really interesting. 6 out of the 7 cysts from this particular sheep shared a completely new, unique haplotype that had never been documented before.
Whoa, a brand new one. That's crazy.
Yeah, it's a pretty significant finding.
Yeah. What are the implications of finding something like that?
So this discovery suggests that the parasite is evolving and potentially spreading at a rate that we might have underestimated before.
Oh, wow. Okay.
It really kind of has important implications for understanding how the parasite's adapting and how it's spreading.
That's a little scary.
Yeah, it definitely raises some questions.
Yeah, for sure.
Uh-huh.
So how do they think that this new haplotype emerged? I noticed the study mentioned a couple of possibilities.
Yeah, so the study proposed 2 potential scenarios.
Okay.
The first scenario suggests that the sheep might have ingested eggs
Okay.
from multiple different parasites,
Uh-huh.
each carrying a slightly different genetic makeup.
Okay.
So that could explain the development of cysts with distinct haplotypes
That makes sense.
within a single host.
So what's the other possibility?
The second scenario involves the possibility of cross-fertilization. Okay.
So in this case, adult tapeworms
Right.
of different genetic lineages could be mating
Okay.
within the intestines of the dog host.
Interesting.
And this mixing of genes could then lead to offspring
Yeah.
with new combinations of genetic material.
Okay.
And these new combinations could explain the diverse haplotypes that were observed in the sheep.
Gotcha. Okay. So both of those sound very plausible.
Yeah.
Do they have any leanings towards one or the other?
Yeah, that's kind of the main question here.
Yeah.
Based on the data that's available in this study,
Yeah.
it's difficult to definitively say which scenario is more probable.
Okay.
So really more research is needed
Gotcha.
to disentangle these possibilities and to pinpoint the primary driver of this genetic diversity.
So this research has really gotten me thinking about the bigger picture. Like, why is it so important to understand the genetic diversity of this parasite? What kind of impact could this have on, sheep and goat health? Yeah, that's a great question. Understanding the genetic diversity of Coenurus cerebralis is crucial for several reasons.
Okay.
First, it can give us valuable insights into the parasite's evolutionary history
Okay.
and how it's adapting to different environments and hosts.
Right.
And this knowledge is essential for developing effective control strategies.
Can you give me an example of how that might work?
Certainly. So let's say we develop a drug that targets a specific gene in the parasite. However, if there's a lot of genetic variation within the parasite population,
Right.
some individuals might have mutations that make them resistant to that drug.
Okay.
So by understanding the genetic diversity, we can develop drugs or treatments that are effective against a wider range of parasites.
So it's kind of like personalized medicine,
Exactly. Yeah.
but for parasites.
It's a tailored approach.
That's really cool.
Yeah.
Are there any other practical implications?
Absolutely. By tracking the spread of different haplotypes, we can gain a better understanding of how the parasite is moving across geographic regions.
Oh, wow.
And this information can be invaluable in identifying higher risk areas
Okay.
and implementing preventative measures to protect livestock.
It's really amazing how much we can glean from a single case.
Right.
The discovery of that new haplotype really makes me wonder if there are even more undiscovered variations out there.
Yeah, that's really the big question here.
Yeah.
And it really underscores the importance of continued research in this area.
Right.
The more we learn about the genetic diversity of this parasite, the better equipped we'll be to manage and control its spread.
Absolutely.
And ultimately protect the health of sheep and goat populations.
This deep dive has been incredibly eye-opening. I feel like this seemingly simple case of a sheep with multiple cysts has actually opened a Pandora's box
It really has.
of questions about this parasite's evolution, adaptation, and spread.
Yeah, and it really seems like we've only just scratched the surface.
Yeah, definitely. So to recap, we've got this unique case of multiple cysts in a single sheep brain, which led to the discovery of a brand new haplotype of this parasite.
Exactly.
And this raises some really fascinating questions
Yeah.
about how it's evolving, adapting, and spreading.
Yeah, it really underscores the importance of ongoing research into the genetic diversity of Coenurus cerebralis.
It leaves me wondering, could this deeper understanding of the parasite's genetics ultimately lead to more effective control strategies in the future?
That's a great question, and one that definitely warrants further investigation.
I'm sure. Well, from Vet Neurojar, keep those minds-inspired hearts light and tails wagging.